In this video segment, we will discuss the details of Phase II Metabolism. The learning objectives for this video are to describe the basic principles of Phase II metabolism, explain how glucuronide, sulfate, and glutathione conjugates are created, compare and contrast Phase II metabolism reactions, and predict Phase II metabolism reactions that will occur at common functional groups of compounds. The substrates for Phase II metabolism can be parent compounds of drugs or toxicants, or products of Phase I metabolism. Phase II reactions use transferase enzymes to attach endogenous polar molecules to the substrate. The three primary enzymes that catalyze Phase II reactions are glucuronylyl transferases, which form glucuronide conjugates, sulfotransferases, which form sulfate conjugates, and glutathione transferases, which form glutathione conjugates. Phase II reactions form highly polar derivatives that are more easily excreted from the body. In most cases, Phase II metabolism makes the compound less biologically active or less toxic. We'll go through the details and examples of the three most common Phase II reactions. The formation of glucuronide conjugates is catalyzed by UDP glucuronylyl transferases, which requires the co-substrate UDP glucuronic acid. UDP stands for uridine diphosphate and is a derivative of the nucleobase uracil. In this reaction, the glucuronic acid from the co-substrate becomes conjugated to the substrate, and uridine diphosphate is released. Glucuronide conjugates are large and polar, and they are primarily excreted through the bile. Glucuronide conjugates are typically formed from carboxylic acids, aromatic amines, aromatic sulfhydryls, and aromatic and aliphatic alcohols. The presence of these functional groups on a molecule makes them good substrates for glucuronidation. Let's look at an example. From the chemical structure of catechol, can you guess where this compound may undergo glucuronidation? Catechol can be glucuronidated at either of the OH groups to form the conjugate product. Now let's discuss sulfation reactions. The formation of sulfate conjugates is catalyzed by sulfotransferases, which require the co-substrate 3-phosphoadenosine 5-phosphosulfate, or PAPS, which transfers a sulfate onto the substrate. Sulfate conjugates are very polar and are primarily excreted in the urine. Substrates for sulfotransferases include aromatic amines and aromatic and aliphatic alcohols. Only primary and secondary alcohols can react with sulfotransferases. This reaction demonstrates the formation of a sulfate conjugate from the aromatic alcohol, phenol. This next reaction shows how a compound such as toluene can undergo both Phase I and Phase II metabolism. Toluene is oxidized to benzyl alcohol by a cytochrome P450 and then conjugated with sulfate by a sulfotransferase to form a product that is easily excreted in urine. Next, we'll go over the third most common Phase II reaction. The formation of glutathione conjugates is catalyzed by glutathione transferases and requires a glutathione co-substrate. Glutathione reacts with strong electrophiles to form conjugates which are further transformed into mercapturic acids, which are primarily excreted through urine. The presence of mercapturic acid in the urine signifies that a reactive compound was formed during metabolism. When drug companies investigate new drugs, this is typically a bad sign for them because the formation of reactive metabolites can often cause cellular damage, especially if the co-substrate glutathione becomes depleted. Glutathione is made up of 3 amino acids, glycine, cysteine, and glutamic acid. The sulfur in cysteine acts as a nucleophile and reacts with strong electrophiles to form a glutathione conjugate. The substrates that glutathione reacts with are often products of Phase I reactions. Glutathione plays an important role in preventing toxicity during metabolism because many of the substrates are highly reactive and could react with essential molecules in cells such as proteins or DNA and cause cellular damage. Let's look at the example of acetaminophen, commonly known as Tylenol. Typically, 60 to 90% of acetaminophen forms glucuronide or sulfate conjugates, but about 5% of the consumed dose will form this reactive metabolite, N-acetylparabenzoquinone amine. This metabolite can react with cellular macromolecules such as proteins and DNA, resulting in cell death and toxicity. However, glutathione can prevent toxicity by forming a conjugate with this reactive metabolite. When you take the recommended dose of Tylenol, the reactive metabolite will not cause toxicity. However, if someone overdoses on Tylenol, they will deplete their glutathione and the reactive metabolite will cause toxicity. It's important to keep in mind that there are species differences in the metabolism of certain compounds. For example, cats have a very low ability to form glucuronide conjugates. This may alter the pharmacokinetics of drugs and toxicants, making some drugs unsafe or some toxicants more harmful for animals. Species differences in metabolism and pharmacokinetics of drugs can also make it difficult to translate research done on animal models to human application. Metabolism is an important pharmacokinetic principle to understand because the rate of metabolism influences the duration and intensity of a chemical compound's action. The type of metabolic reactions determines how the compound will be eliminated, and metabolism can contribute to the formation of reactive and toxic metabolites. In the next video segment, we will discuss the elimination of drugs and toxicants from the body. To test your knowledge of Phase II metabolism, you should be able to answer the following questions.